thank you all for joining us today. It's morning, of course, here in the US. To those of you who just finished the long weekend here, hopefully you had a good Memorial Day weekend and better weather than we did here in Utah. I'm looking out my window and seeing snow across the valley. I'm sure it's not like that in most of the world. So this is the second of a series of webinar seminars that we've been trying to organize through the Consortium for ECGI, in which we bring leading researchers together in the setting of the consortium to try and explore some of the different aspects and utility and applications of ECGI imaging, both the technical all the way through to the translational. And I think we have a nice spectrum even within the two speakers we have today. And there are two main speakers, and we've asked Steve Niederer also to give a brief introduction of what his lab is up to overall, and we'll get that in just a sec. So as I say, we have two major talks today. One very much, I think, focused on clinical applications of ECGI, and I think this is always of interest. This is where we want to go with this technology. So these are always very relevant talks to hear. And then the second one is opening up sort of a new approach to broadly modeling and including ECGI, and that's what Natalia Trayanova will tell us about in the concept of neural networks and machine learning as applied to our little disciplines. So the speakers that we have today, like I say, is Steve Niederer. Professor Niederer is a professor of the School of Biomedical Engineering and Imaging Sciences at King's College, London. He's I feel really excited today because I feel like I'm sharing the show with two rock stars and a new rock star in the making. So Steve is definitely a rock star in our field. As you'll hear, he's involved in a number of different projects, and we've asked him just to give an overview of what all the lab is up to before we then switch to his former PhD student, now a postdoc in the lab, Marina Strocchi. And Marina, does electromechanical modeling, which is a big part of what Steve's lab is engaged in, but also works directly with clinicians and helping them use and apply and interpret ECGI. So I think this will be really relevant. The second talk will be with Natalia Trayanova, and she's the Murray B. Sachs Professor of Biomedical Engineering and Medicine. She's appointed in both departments and very involved also in this translational space, involving imaging, modeling, patient-specific approaches to try and understand cardiac arrhythmias and predict where they might occur, and even guide therapies and ablation therapies in the context of ventricular and atrial arrhythmias both. So with no further ado, let's move forward. So with that, I'll hand over first to Steve to give a brief introduction of what his lab is up to, and then he'll pass it on to Marina.
There we go. Thanks very much for the introduction, Rob. So this is a very quick introduction. When Rob asked me to give a presentation on ECGI, I said that it would probably be best to have the person who works with ECGI give the talk as opposed to me. So this is just a summary of the work in the lab. So within the group, we are really thinking about how we can use computational models to inform heart failure or how we treat the heart. And this was a nice publication that one of my students, Christopher, put together for kind of younger children about what we're trying to do. And I think it's very useful to convey what we're trying to do to cardiologists who have a similar mindset. And that is that we're trying to make a computer model of the heart that doctors can effectively play with, run a bunch of tests on, and then try and work out how they can make the heart better through simulation, and then apply what they've learned to the patients. And so what we're doing for building these models is we treat the heart as a multi-scale and a multi-physics system. And so we start thinking about the heart with electrical activation, propagating over the right atria, over the left atria, before activating the left and right ventricles. And this electrical signal that we see propagating over that whole heart is manifested in our cellular models of electrophysiology, which capture how ions move in and out of the cell across the cell membrane, leading to an electrical signal at the cell scale. That electrical signal then leads to a release of calcium ions. So we have mathematical models capturing the sarcoplasmic reticulum and intracellular calcium regulation, which then go on to how calcium ions bind to sarcomeres, leading to the generation of tension and contraction in the myocyte. And these cell models that we have are then embedded within a tissue model of the tissue matrix, capturing the preferred orientation for stiffness, electrophysiology, as well as for the orientation of contraction. We can then go on to combine these contraction models with models of anatomy, capturing the deformation. So we can then simulate the pumping of the heart. And finally, we can then link these models of deformation through to computational fluid dynamic simulations, simulating the fluids as they move through the different changes of the heart. And this is all done using a portfolio of software, both the CMERGE app application within our group, as well as CARP from Gernot Planck's group. We use SIG3D, which I've labeled there from Utah, as well as kind of a portfolio of other open source tools and machine learning technology for calibrating the models. And so this, when we put all this together, we end up with a dashboard representing how the whole heart can simulate, capturing electrophysiological activation on the right, but also capturing the different volumes in the aorta and the ventricles, and the right and left side of the heart, kind of providing us with these 4-chamber cardiac electromechanic models that we're it's kind of the direction of travel for us in the simulation space. When we think about how we're applying this in the clinic, we look at putting endocardial, one of the things we look at is CRT cases. These are patients who are having a pacemaker device implanted, and we want to know where to put the electrode in this patient's heart. This is a leadless endocardial electrode. And we're also looking at where we'd like to put a transmitter, which is the ultrasound transmitter, which is providing energy for this particular device. The specifics of this aren't terribly important for the application. So we're looking at where we can put the electrode. And what we're currently doing is taking ECG, just standard 12-lead ECG and CT images, and using these images to capture anatomy. So looking for regions of thin myocardium where we wouldn't want to put the electrode, looking at capturing motion in the heart. So we look for regions which are late contracting, looking at simulating electrical activation so we can predict where to put the electrode, and measuring perfusion in these patients. We can then work out where to target these electrodes from this combination of anatomy and simulation-based predictions, look where to put the electrode, and then put those targets and overlay them on top of the fluoroscopy images during the procedure. And for us, this is really important because we want to take information out of the clinic and then look at how we can effectively put it back into the clinic, and then use that to guide therapy for put this in front of the cardiologist so they can then work out where to place the electrodes during the procedure. But what we've really found is a limitation, and this is a nice segue into Marina's talk, is that we're currently using 12-lead ECG, and this provides a relatively limited representation of the electrophysiology of the heart. Now, we can go into the patient's heart and make invasive electrical anatomical maps, which we do for our atrial work, but in many ways, this is too late. The decision's been made, the procedure is going to be going forward. And so we really see that ECGI is a really nice way of calibrating these models and making better models for making predictions. And with that, I pass over to Marina for her section. So I apologize if I talk too long.
Thanks, Steve. That was great. All right. Marina, the floor is yours.
Marina, you're muted.
Can you hear me now? Right. Sorry. Okay, sorry. Thank you for giving me the opportunity to present my work here. So I'm just going to start my presentation by giving you an introduction of basically how we analyze ECGI data, starting from the type of data we deal with up to what we extract from all the data we get from the clinicians. And then I'm going to show you 3 studies where we applied this analysis in collaboration with different clinical groups. In the, as I'm sure all of you know, during ECGI, the patient has a VAS. In particular, the data I'm going to show you are all with CardioInsight, the mapping VAS from Medtronic, which has 252 electrodes, I believe. After data acquisition, the patient undergoes a CT, and the clinicians segment the torso and the epicardium, in this case of the ventricles. And then the torso signals acquired during the acquisition together with the geometries are fed into the Medtronic software to get the epicardial signal of the ventricles. And this is where basically I start my data analysis. So we've got the unipolar electrograms on the epicardium of the ventricles over time. And an example, you can see the are the black signals. For activation and repolarization time computations, we need to isolate the QRS and the T waves, which you can see windowed here in red and blue. Then we filter the data and we compute activation and repolarization times. So activation times are computed as the time of the most negative downslope during the QRS duration, while the repolarization time is computed as the time of the maximum upslope during the T wave. And then we can compute the activation recovery interval, or ARI, as the difference between repolarization and activation times, which is a surrogate for action potential duration, which is important when we want to look at arrhythmias risks. So sometimes due to poor quality of the signals, we end up with areas where we can't compute activation or repolarization. Here you're looking at a map of activation times with a gray area where we can't see the where we can't compute the activation time. So to kind of deal with this, we use the Gaussian process manifold interpolation, the code for which is available online on GitHub. So if you're interested, I'm happy to talk about that a bit more. And this basically gives us interpolated maps where we can basically extract the information we're missing. And most importantly, we also get uncertainty. So we also know how confident we are about the interpolation we're using. So we tested if this interpolation could be trusted before using it in our pipeline. So we took a beat for a left bundle branch block patient for which we could compute activation and repolarization times at every node. This was a very good quality beat. So here you're looking at the maps we extracted from the beat, activation, repolarization on the right and on the left and the right. Then we removed 10, 20, up to 70% of the values, and we interpolated every time, and we compared the difference between the interpolation and the true value, let's say, and we computed the error and the uncertainty on the prediction. And here you're looking at the results. Activation repolarization results ensured in pink and green. On the left, you have the absolute error, and on the right, you have the uncertainty. And you can see that as we remove more points, our error is not affected that much, but the uncertainty increases, which makes sense because you are giving the interpolation more sparse information, and so the uncertainty on the predictions will increase. And what's interesting is to look also at where the biggest error happens, basically. So here you're looking at activation and repolarization maps with the spheres representing the points we have removed, so where the error and the uncertainty are evaluated, and the size of the sphere is proportional to this, how big the error is. And you can see that the error is bigger where we have crowded isolines, and that is because the interpolation assumes a certain level of smoothness of the data, and so the interpolation is not able to capture very sharp changes in activation and repolarization. But overall, we had quite a small error, so we were quite happy with this, and we decided to use the interpolation on our in our pipeline. So now I'm going to move on to applications. So the first one was published in the Journal of Electrocardiology in collaboration with Dr. Mark Elliott, and basically was looking at cardiac resynchronization therapy. So as Steve mentioned, cardiac resynchronization therapy, or CRT, is effective at reducing activation times of the heart and this aims to improve electrical synchrony and therefore the heart pumping function. However, there are unknown effects of CRT on repolarization times, and this is what the study looks at basically. So what are the effects of CRT on repolarization? So we had 11 heart failure patients undergoing CRT. So data acquisition was done 1 day after CRT implantation and 6 months after. And this is because repolarization remodeling or electrophysiology remodeling is thought to happen mostly over time. And so that's why the acquisition was done also 6 months after. And we analyzed 3 beats at baseline and 3 beats during pacing. And we computed activation, repolarization times, as I explained before. And we also looked at the activation recovery interval, which is, again, a surrogate for action potential duration. So the clinicians are interested in metrics, in simple numbers, basically, because they want to draw a conclusion about the overall function of the heart. And so we have a way of separating the left ventricle, the right ventricle, and the septum. And we also exclude the base from when we compute any sort of response when dealing with CRT. And that is because the base of the ventricles is thought not to contribute too much to heart function. And so it's always excluded when computing metrics. And in particular for this study, we focused on the left ventricle, which you can see in blue. So we looked at the dispersion of action potential duration in the left ventricle, because that is an indication of how the patient is at risk for arrhythmias. So you can see that at baseline and after CRT, the dispersion of ARI increases, and it increases even more in a significant way after 6 months during pacing. And in particular, this effect was even more pronounced in patients that did not respond to therapy, where no response was determined looking at how much smaller the end systolic volume of the patient was. And so basically, this study indicates that CRT can induce long-term electrical remodeling, and this can increase the patient's risk to arrhythmias during pacing. So the second application that I'm going to show you is always about CRT, but to kind of explain, to kind of give you a background of the study, I need to explain a bit more about CRT. And if you're interested in more details, we published an abstract on the Heart Rhythm Society meeting in 2022, so this year, and it's a poster presentation by Dr. Mark Elias. So as I mentioned before, CRT aims at restoring electrical synchrony in patients who have a dyssynchrony between the right and the left ventricle. And this is done by pacing the heart at the right ventricular apex and at the left ventricular epicardium. And although this works in most patients, there are still some of them who do not respond to the therapy. And that is why clinicians are always researching better and new ways to delivering CRT in order to reduce the proportion of non-responders. And 3 examples of these are endocardial pacing, his bundle pacing, and left bundle pacing. So during endocardial pacing, the left ventricle is paced at the endocardium rather than the epicardium. And that's thought to be better because we have easier access to fast conducting tissue in the endocardium. So it was shown to lead to better response. And his bundle pacing and left bundle pacing are examples of conduction system pacing, where we pace the fast conducting system of the ventricles. And so they are, in theory, the best way to pace the heart, because we have the opportunity to restore electrical synchrony as if the block never happened. And this study kind of started from a study we published a couple of years ago on heart rhythm. That was a computational modeling study looking at his bundle pacing, left bundle pacing, endocardial pacing, and standard CRT. And we compared those pacing modalities with computational electrophysiology. And we want to see basically if the findings of that study with computational modeling are true in patients, and so we use the CGI to do this. So again, we have heart failure patients, this time 7 of them, undergoing CRT. These patients underwent standard CRT implantation, so LV epicardial lead and RV endocardial apical lead. And then they also underwent temporary endocardial CRT, his bundle pacing and left bundle pacing. And in terms of data analysis, we have 3 beats as baseline and 3 beats during each pacing modality. In this case, we only looked at activation, and the way we compute metrics is always the same as before. So we separate left ventricle, right ventricle septum, and we exclude the base. So here you have an example of the maps, of activation maps we produced with our data analysis. So in red, you're looking at early activation, and in blue, late activation. So these are maps from a left bundle branch block patient. So that's why you're seeing very quick activation of the right ventricle and late activation of the left ventricle. And then with standard CRT, the activation was better. So you don't have any more blue areas in the ventricles. That is because activation is shorter. But activation was even shorted with endocardial pacing, his bundle pacing, and left bundle pacing. And this was true for all patients. So on average, we looked at the relative change in left ventricular activation time and left ventricular desynchronous index, which is the standard deviation of activation times. And here we are looking at reduction between baseline to pacing. And you can see that the reduction in activation time we achieve with standard CRT is smaller compared to the reduction we get with all other 3 pacing modalities. And so this basically showed that novel pacing techniques have the potential to reduce activation times more than standard CRT, and so they are likely to lead to less non-responders. And this was in agreement with our computational study I mentioned before. So the last application I'm showing you today is about Brugada syndrome patients. So this is in collaboration with the University of Sydney, but the study is still underway, so I'm unable to share the final results. But I'm gonna show you some examples of the data. So Brugada syndrome is an inherited disorder. It is rare, but it's thought to cause 20% of deaths in patients with structurally normal hearts. And because it's so rare, there's still disagreement about the mechanisms underlying Brugada syndrome. One hypothesis is that the RV outflow tract activates late, causing heterogeneity in activation times in the right ventricle. And the other hypothesis is that the right ventricular repolarization is abnormal instead. And we wanted to use a CGI to kind of test these 2 hypotheses and look at activation and repolarization times in Brugada syndrome patients. So the reason why I'm showing these results, these data, is because they highlight kind of some difficulties we had while analyzing the CGI data, which I think are quite interesting. So in Brugada syndrome patients in general, we expect to see, or we expect it's likely that the patient has late right ventricular outflow tract late activation. And here you're looking at activation maps for one of the patients with 6 baseline beats. And in blue, you have late activated regions. And you can see that we see late activation for a few beats, but not all of them. And these are the electrograms in the area where we couldn't detect late activation, but where we would expect it to be. And you can see the electrograms in black and the detected activation time in red. So if we focus on only one electrogram, so here you have with the electrogram at the top, and at the bottom you have the derivative. So the activation time is normally is detected as the time of the most negative downslope during the QRS. And basically the first downslope is where we detect activation, because that is steepest compared to the other one that is indicated by the arrow. However, if we wanted to see how the activation times would change, or the maps would change, if instead we take the latest steep downslope during the QRS, and these basically are how the maps change. So you can see the right ventricular outflow tract in this case is activated late for all beats. And so this is more what the clinicians expected for this sort of patients. And so this just highlights how important it is to always have a back and forth with the clinicians, and also to keep in mind which sort of patients you're working on when dealing with ECGI data. So just to conclude, I showed you our methods for ECGI analysis, starting from the epicardial signals. We can compute activation of repolarizations, and I mentioned how we deal with noisy or low amplitude signals with the interpolation method. And then I showed you 3 studies where we applied this analysis. The first one looked at repolarization during CRT, and I showed you how we reached the conclusion that CRT can increase dispersion of APD in the heart, leading to increased risks of arrhythmias. The second study I showed you was always about CRT, but focusing on activation and looking at different ways of delivering CRT. And the last one was an example about a Brugada syndrome patient, where changing the way we computed activation changed also the activation maps, showing challenges in ECGI data analysis. So I would just like to end my presentation by thanking the clinicians who acquired data. So Dr. Mark Elliott and Professor Aldo Rinaldi at King's College London, but also Dr. Julia Isbister and Dr. Raymond Tsai, who acquired the data at the University of Sydney from Brugada syndrome patients, and also everyone in Professor Steve Niederer group, which is where I'm doing my postdoc now. And I would also like to thank you for your attention, and I'm happy to take questions.
Great, thank you, Marina. That was excellent. Amazing density and breadth of the topics that you've been covering. This is really exciting. So I have plenty of questions, but I will allow a little bit of time maybe now. I'd love to push questions a little bit to the end so we can bring both speakers into that. And if there's nothing super pressing, the one question I'll ask you to think about maybe at this point, because I want to hear also from Natalia about this. You mentioned it several times, and I think with this engineering group, it's an incredibly relevant question to help us, or through your experiences, describe how you interact with the clinicians. How do you present this sort of very technical approach to the clinicians in a healthy way? You showed a lovely example in the Brugada study of where you had to iterate with the clinicians and refine your interpretation of the results. And I'd love to hear both of you talk about that maybe at the end of the seminar. So perhaps we can, looks like nothing else is too pressing. I don't see anything in the chat or hand up. So let's move on to the second speaker, and then try and have some time at the end to bring you both back on stage and have some Q&A at that point. So with that, I'll pass. Thank you again, Marina. That was excellent. And I'll pass the baton on to Natalia Trayanova, my dear friend and colleague. I was saying before you joined, Natalia, that I feel incredibly lucky today to be sharing the stage with 2 of the rock stars in our field. And I think you certainly qualify. So we're looking forward to hearing your presentation on some of these new AI approaches, machine learning approaches in our field.
Thank you, Rob. Thank you all very much for inviting me. I'm gonna see how this works. I hope you see it in as it's supposed to be. So again, wonderful. Thank you, Jake. So normally my students and postdocs do the work and then I present the results in an overview form. So that's what I'm going to do today. I'm going to share with you our newest approaches that we have developed over the last couple years, 3 years, and they are all basically what we do is extremely clinically oriented, and you'll see evidence of that. We work very close with clinicians, and as Rob said, we can discuss how that happens in our hands. So what our goals are in general in my lab. I also direct a what we call the Advanced Center. This is an alliance for cardiovascular diagnostic and treatment innovation, and we work very synergistically with imaging people and clinicians. So our goal in my lab is to develop these sort of virtual hearts, digital heart technologies for specific clinical applications. And typically we tailor them to the application. And of course, we are putting a significant emphasis on AI approaches these days. Another very important component of what we do is we validate the usefulness of our technology with clinical outcome. And here the emphasis is on the fact that we do not need to have the best models. They do not have to be absolutely correct or incorporate every physiological detail, but they have to be clinically useful. That's what determines whether what we do is successful. We would like to work towards democratizing patient healthcare delivery. We want to incorporate our approaches with clinical workflow, and we are doing that, and then have prospective studies and trial and ensure standardization across clinical centers. So I'm gonna focus on two important components of our work. These are the two research avenues that we have been focusing for a while. It is the diagnostics or prognostics, predicting particularly risk of sudden cardiac death. And the other is the treatment, the ablation guidance for atrial and ventricular arrhythmias. Today, for the lack of time, I'll just show you our atrial approaches. So I want to jump directly into one of the newest approaches that we have done for predicting risk of sudden cardiac death in patients. Initially, we started with patients with ischemic cardiomyopathy, but as we moved to more complex diseases, it became clear that we cannot just be building computational models of imaging as we have doing before. It is very important to incorporate the clinical data. So the way we modified our standard pipeline for creating computational model from imaging data, having electrophysiological properties, and then we assessing arrhythmia propensity in the digital substrate, we are now combining that with machine learning approaches on both simulation and clinical data to stratify risk of sudden cardiac death. So I want to show you a quite complex disease called cardiac sarcoidosis. It's an inflammatory disease, and what it is characterized with is severe inflammation and also interstitial fibrosis. So typically PET scans are used to visualize the inflammation, while LG-MRI visualize the enhancement which corresponds to the regions of fibrosis. So we embarked on conducting computational modeling and artificial intelligence in that, and this is the paper. It was published in the early fall in Science Advances. The first author is Julie Shade, who you see here recently graduated and now works in a startup. So it was an amazing work that she did. Our cohort consisted of 45 patients that both had LG and PET scans that were acquired about the same time, and so therefore we had simultaneous visualization or into different imaging modalities of fibrosis and inflammation, and 16 of these patients had clinical VT. So here is what we did first. We created for the first time this MRI PET fusion model. So from the two scans, we combined them and created a computational model, or at least first geometrical model, that incorporates inflammation, fibrosis, as well as overlap of fibrosis and inflammation. And these three regions have really distinct properties. This is a view of one of the hearts that we reconstructed. And then we populate these with different electrophysiological properties based on the literature. We very hard try to gather all the literature available for these electrophysiological properties. Once you create the models, the first thing is validation with clinical studies. So separate from the cohort of 45 patients on which we are doing the prediction, and the prediction is blind to the clinical outcome. Separate from that, we validated the model with in patients who are undergoing ablation with cardiac sarcoidosis, and then we compared the predicted ablation targets in our models and in the clinic. So you can see here, for instance, on the bottom is one patient. You can see the red dots are the ablation targets, and this is the predicted in our simulation, the re-entrant circuit in exactly where the ablation was. And this is another, this patient had a substrate ablation, so you know, it is a larger region where it was ablated, but also we had a very good coincidence with these ablation targets. So in the paper we go in detail about the validation study. So once we validated the models, here is what our study consisted of. So we have a mechanistic component model in modeling when we combine PET, MRI, EP, all in the model, and then we predict arrhythmogenic propensity of the substrate. Then we used also all the imaging data, clinical data, output of all simulations. All that went into a random forest feature selection, and from there into a risk predictor to predict the risk of sudden cardiac death on these patients. We called the approach CHI, computational heart and artificial intelligence risk predictor. So basically the most important features are selected, and then the predictor predicts that risk. So I want to show you all the results, like directly in the results. So here are our results. So you can see here, so we had a validation and testing, cross-validation and testing. You can see here the confidence intervals. We did many repeat cross-validations. So our sensitivity and specificity are quite high, both in cross-validation and test, as you can see. The positive predictive value, negative, and of course, importantly, the area under the receiver operating curve, which was quite high. Now, compare that with what is currently in the clinic. So this is data on the same patients acquired in the clinic. So these patients, ejection fraction less than 35% was used to predict. You can see sensitivity, specificity, positive predictive value, negative predictive value. Fibrosis as a predictor for risk of sudden cardiac death, low specificity. Abnormal uptake on PET, also low specificity. We have a much more balanced specificity sensitivity. As a matter of fact, balanced sensitivity and specificity was one of the hyperparameters on which the predictor was optimized. So here we demonstrate that when you combine computational modeling, which is mechanistic and can give us an idea what is the propensity of the substrate, and combine with these factors that promote arrhythmogenesis, that are clinical factors, we were able to arrive at a really good predictor for sudden cardiac death here. What is important also is that we had a small cohort, but it worked very well. Why? Because the machine learning algorithm does not have to, it doesn't need to learn from the electrophysiological simulations output. They incorporate laws of physics, knowledge of physiology. So some of these features that get extracted, they carry that, so they don't have to be learned. So it worked very well on a small data cohort. So now again, I entitled my talk as a combination of computational modeling and artificial intelligence. So this was a true combination. Now I want to show you something that is pure AI, and it's actually a very sophisticated AI that we did in our most recent paper that was published less than a month ago. So in this paper, we developed something that we called SCAR, which is a sudden cardiac death predictor. This is a risk predictor of sudden cardiac death, but it predicts actually the time to sudden cardiac death. What's important, it's not a yes or no predictor. It predicts when the patient is going to have an episode. This was a very complex and convoluted and really study that involved different clinical groups and a lot of resource to develop, in which we developed a deep learning network straight on raw images. And I will come back to this point. These are raw, unsegmented MRI images, LG MRI images. So we developed a neural network, deep learning network on those images. And then we had another neural network on clinical covariates. And all that was embedded in survival analysis. So we can actually predict personalized probability distribution of time of sudden cardiac death. So we construct these individual patient survival curves. So in this slide, I really emphasize what are these important components of this new technology. We applied that in patients with ischemic heart disease, and we had a cohort on which we trained, and a completely separate external cohort from many hospitals on which we did the external validation. So it was termed survival study of cardiac arrhythmia risk, and it's a good pun to be called SCAR. So we estimate the individual patient's time to sudden cardiac death. Importantly, it's learning from raw clinical images, and the probability that the time when the event occur, offer probabilities up to 10 years. So we can predict who is going to have an episode in the range between now and 10 years. And what is important also in this methodology is every prediction carries uncertainty in it, and it's learned from the data itself. So this is the construction of the predictor. We had an internal cohort that is administered at Hopkins, but it's actually 3 hospitals. 1 in Delaware and 2 in the state of Maryland. And these were all patients with low ejection fraction. Now, the external test cohort was a different patient group. These patients were with moderate decrease in ejection fraction. So if we perform well on that cohort, that would be remarkable because it's completely unseen. These patients were collected, the data were collected from 6 different hospitals across the country. It's part of the predetermined trial. So those were our patient population. So, and the second neural network learned on the clinical covariate data, and those are all the parameters that went into the deep learning network. It's important that the clinical networks went also into a deep learning network because we are able to take into account the nonlinear dependencies that exist in the data set. Okay, so here is the overall performance. So we did a lot of different performance indicators. This is, you can see here on the internal data set, and that's the external data set. So this is the area under the receiver operated curve. You can see in a very different cohort, completely different, it still performed quite well. And this is the average over 10 years. What is shown here is the concordant index. You can see it stays quite high over the 10 years. Yeah, so this is sort of summarizes the overall performance. You can take a look at the paper, you can see we show how the uncertainties, you can visualize the uncertainties in every prediction. But another important step that we did also is we tried to have, because those are 2 deep learning networks that are combined into this survival analysis, we also try to have some sort of interpretability of the outputs. So we use here a new technique called gradient activation maps, in which you sort of try to lift the hood under the deep learning and figure out what are the parameters that had the biggest contribution and what had the least contribution. So here is in imaging. So this was a patient that did not have a risk of sudden cardiac death, and you can see if it is blue, means nothing contributed here to learning anything from these images. While here, this is a patient, they are regions from which the neural network considered really important. And when we compared it the scar distribution, they were not one-to-one. So it learns from certain things, including, of course, the distribution, but it's not only it. And these are some of the clinical covariates that were the most important. So red is where it contributed the most. Okay, so here, so those are the most important. So why we think this was so awesome? Because it offers these accurate probabilities at all times up to 10 years. It captures complex imaging and non-imaging dependencies. It works on raw images. You don't have, we were lazy. We didn't want to be segmenting like hundreds of patients, so that's why we went for that. It really works very well on these imaging data. It predicts entire probability distributions, allowing for uncertainties in predictions to be learned from the data and are patient-specific. So we believe that it can become a personalized, groundbreaking strategy for non-invasively predicting risk of sudden cardiac death. This is the paper. This is the first author. It got published in Nature Cardiovascular Research. 2 weeks after the paper was published, Dan, who was the first author, got hired to be the vice president for data science in Goldman Sachs. So it tells you how much his machine learning skills were appreciated. The paper was on the cover of the magazine. You can see our little neural network here on the slices from which we learn. But the pinnacle of my life is that the paper was mentioned on Saturday Night Live, which is a very famous comedy show in the US. And this is Colin Jost, that was on April 16, who's the husband of Scarlett Johansson, by the way. So this is the pinnacle of my life. I think we have arrived. There is nothing more we can do once we were mentioned by that show. So this is the story with this paper. Now I want to move. Move it. I don't want to. Okay. The other outcome of this paper that I was invited to write a editorial with Eric Topol, who's like the god in medicine for the Lancet. So together with the Saturday Night Live show meant a lot for us. Anyways, I want to move into the last few slides here and show you what we do for treatment. So this was diagnostic and what we do for treatment. I don't have time to go through the AI approaches there, but I want to show you something that I consider probably one of the most important things that we are doing in my lab. So this is a paper that was published in 2019, and this is a computationally guided, personalized, targeted ablation of persistent atrial fibrillation. Now, this is a prospective study entirely. There is nothing retrospective. We predict where to ablate. They ablate it in the clinic. We are part of the procedure. We are there, then ablate the patient, and they were followed for 2 years. And then we enrolled 10 patients. The approach was called OPTIMA, optimal target identification via modeling of arrhythmogenesis. Here is our pipeline. They're patients with persistent AF. We do pre-ablation MRI, segment the MRI, do 3D reconstruction, predict the ablation targets. We do not stop there. Then we do an important part, which is we do a mock-up of the actual clinical procedure. We perform virtual ablations in the model and then identify whether after you ablate, whether there are new targets that occur, and we repeat that. And finally, we export that into the electroanatomical mapping system. We reverse engineered it. Now we can import in any electroanatomical system our predictions. So this is the integration of our images with CARTO. You can see we go through the MRA. It's different for the left and right atrium. And finally, this is what the physician is going to see on the screen during the ablation procedure. So as I said, we enrolled 10 patients, directly prospectively ablated. This is one of our patients. It's 17 years old. Most of them had a previous ablation, and a lot of them had a pretty high CHA2DS2-VASc score. They had a lot of comorbidities. Nonetheless, we proceeded with that. Basically, we took all the patients that no other clinical trial wanted, but so we did the ablation in those. So this is our patient. This patient had, had several targets. So you can see one of our targets is around here. Here is another one. I think there were 3 in the left atrium. What is important in our approach is that we are predicting both left and right. The right is also very important. There are a number of targets in the right atrium. So those are bi-chamber models. This is, whoops. This is another patient. This was the only first-time procedure. So you can see him here, and this is the predicted sort of how the rotational activity looks like. And what you see here is the ablation. So this was in the right atrium, right here. That's where we predicted. And after that, they do PVI. So the reason we do first our targets, then they check whether there is organization of the activity after we hit the target. So here is the outcome of these 10 patients. Those are the days for which they were followed. And normally for persistent AF patients, the outcome of ablation, the recurrence, is about 60% of the patients have recurrence. And you can see here that we have only 1 patient that came back. It was for a flutter, much simpler arrhythmia, and so that's our outcome. Now, what are the conceptual advantages of what we are doing here? So we know exactly where to ablate in the fibrotic substrate. The final ablation set includes emergent targets for ablation as well, and we are hoping that that approach could decrease and even eliminate redo procedures. So what happens after that is we went to the FDA and said, well, should we do a clinical trial on that? And so I did supply, I applied for that, and the FDA approved a 160 patient randomized clinical trial to determine the utility of this approach, in which it is patients are randomized to PVI or PVI plus Optima. It's a single center trial. It's only at Hopkins. So this right here is not 13. This is an old slide. We are at 20. We just ablated 2 days ago another patient. We have done 20 patients. We hired Eugene Kolmovsky of Rob's institution. He is doing amazing images for Optima. All the new patients he's been with us for a year now. He does all the scans. It's amazing. I've never seen such beautiful scans of the atria, so for both right and left. In order to process that faster, we developed a deep learning network for atrial segmentation, both left and right. We also do have a deep learning network for ventricular, but here we are using for both atria our own deep learning segmentation network. We are actually participating in the MICI challenge for atrial segmentation as well. So that's where we stand with that. And for us, it's a huge deal. I have a team who's working on the clinical trial, and all the clinicians are extraordinarily involved. So these are some of our Optima patients. I don't want to report anything because we haven't processed anything. But to conclude my talk, I think the take-home messages from everything is that personalized mechanistic computational approaches can be synergistically combined with AI to both broaden patient data input and offer explainability. And they can be either by themselves or combined. They can be a major tools in precision cardiology. And I want to thank anybody who's participated in that. I have to say in concluding this that we have discussed on many occasions, how we, in some of the approaches, we can combine ECGI input as well. So for me, it is really, this is one pathway that we have taken, but there are many ways to sort of to band together in making these developments, both with artificial intelligence, computational modeling, and any experimental recording methodologies or clinical recording methodologies that could be there to help our patients. So with that, thank you very much for the attention, and I'm happy to answer any questions that you might have.
Great, thank you, Natalia. As usual, amazing, awesome, enlightening, and motivating, hopefully, for everybody. Otherwise, I'll start with one while people are thinking about it. Maybe let Natalia catch her breath and go back to Marina and see if she has an answer to that question I posed earlier of, you know, what is the interaction with the clinicians like? How do you, you know, introduce them to this technology, convince them of its utility, and deal with their natural and probably healthy skepticism?
So in these in the examples I showed, it was the clinicians who were interested in getting the analysis done. So I think we started from a favorable position. But in general, I think anytime I start a collaboration about an ECGI project, I think the first thing to do is like establish what the clinicians' expectations are, because they don't know how the data analysis happens. But I try as much as possible to kind of have always a back and forth, update them, and be clear about what can be done and what cannot be done, and how this is dependent on the signals. So like one of the examples was the Brugada syndrome patients, where the data didn't really show what they expected. And in this case, we found a reason, which was nice, but that's not always the case. So yeah, I think that there is a lot of room for improvement in repolarization analysis, especially because activation is much easier to deal with. And so that I think it's less likely to lead to unexpected results. Repolarization, there is a lot of room for improvements. At the University of Bordeaux at Lyric, there is Laura Bear. She's using machine learning to improve repolarization times computation. So I think that AI can help in that respect, because it can deal with weird signals that otherwise would need to be checked, for instance, one by one, which is pretty much an impossible task. So I think that combining data analysis we are doing with AI at some point would also be a great approach. But in general, the clinicians, yeah, you need to kind of always involve them in the analysis you're doing. That is very important. Do not assume that they will just treat it as a black box, because they don't most of the times, in my experience at least. So yeah, always keep them in the loop as much as possible.
Yeah, excellent. You know, these seem like fundamentally different approaches, you know, the ECGI signal based, because it delivers something to them that, you know, is similar to what they're used to seeing. So when they see an abnormal result and you can show them an electrogram, they can say, oh, electrogram, I get it. Let's interpret this. Let's think about this. But then when I think, and this is sort of where I'd love both of you to go, is, but AI is truly a black box, you know, for most of everybody but the real core experts. And so how do you find them more accepting of the black box that nobody pretends to understand, the AI, because AI is popular and you know, there's sort of a nice love affair emerging, or how do they respond to those two different types of data, you know, relative to each other?
Are you asking Marina or me, or both?
Well, both. Let's maybe let Marina answer first, and I'd love to hear your take on this too, Natalia.
Yeah, I think that it depends on the clinician you're working with a lot. Many of the clinicians we work with, I think we are super lucky, and they are very open to new technologies and AI in general, computational modeling as well. But I know that it's not always the case. But I think I've been pretty lucky so far. So maybe Natalia is more experienced
Yeah.
with that.
Okay, Natalia, how would you answer the question?
I mean, I don't even ask, basically the way I interact with the clinicians is we've had so it's very important that we have a long history. The fact that they did a prospective study, I mean, look, this is worse than AI actually, because they are ablating on targets that I am predicting and they have no idea how I'm predicting them. So, I can explain how many times how I'm simulating. It doesn't make difference, right? So they did already that. So to me, AI is peanuts as compared to what they did with the prospective ablation. The fact that they put their necks on the line to do an FDA approved study, clinical trial, and one of the clinicians, this is a massive undertaking, because you got to understand there are HIPAA walls. I do not have access to patient data. I'm an engineer. I'm not an MD. So I am leading the whole project on outside of the HIPAA wall, right? I have no access. And so I have clinicians who are willing to undertake all that. I have to hire my own clinical coordinator who coordinates on the other side of the HIPAA wall. And so all these logistics. So once you do that, telling them you're going to do an AI, it's like, okay, sure, we're doing AI, right? The other was much more sophisticated and more organizationally. Also, in terms of AI, they have the trust in you, in your capabilities, I would say. They have worked with us for a while. They are co-authors on every Nature or Science paper that I have written, on any other paper, right? They are there. I am helping their career a lot as well. I am the one who writes the grants. So in a way, I've become this sort of a hub for them. They would come to us, but very often they do not come to us with a project. That's a little different from Marina. Some do, sometimes we do, but most of the time we think differently and we want to say, I want to do this. They cannot tell me, can we do an AI on this data? Because they have no idea what an AI is. So I decide what I want to do a project on. And then I go to them and I say, do you have that kind of data? If you don't, I hire this clinical coordinator who looks at the patient data and extracts for me data that got anonymized after that and sent to me. So I've worked out that channel where they don't even have to give us an idea of what we are doing. We can say, can we do that project? I think it will be useful. They're very much on board, much on board with everything. They just like to see results. They like to see papers published, grants brought. Most importantly though, is they would like to see their patients doing better. And if you can show that, like the clinical study showed that, that's why they were on board with the FDA approved clinical trial. So it's all a combination of those things.
Right. I mean, I can totally see that with the clinicians you know well. I mean, I know what this is like. You have that trusting relationship you've built up over years in your own institution. What is it like when you go to Heart Rhythm Society or or ERA and present these results to a broader body of clinicians? Do you sense their skepticism? Are they dying to try? Are they running up to you and saying, please, show me how to use this in my own patients? What's the response of the broader?
The latter, actually. I have not gone to any engineering meeting in a very, very long time. Most of the time I presented clinical meetings, and every time, and not very often, I'm the only engineer. They all are very interested. If they have skepticism, they have not expressed it to me. I'm sure they are skeptics, always there will be. But majority of the people after a talk would just come to me and say, oh, can we try? And I'm like, I can't. I'm gonna do my clinical trial. What if it is worse? I can't. It's approved by the FDA in a single center. I'm gonna go on with that until I prove. For the AI approach is also so many. The other thing is patients contact me, or individual patients, and I'm not an MD. After that paper with the AI, the last one, oh my God, I got so many patients and I don't even know what to tell them. So it's a lot of interest. I think I get a lot of interest at HRS was a lot of interest. Typically the clinical meetings have a lot of interest. clinicians like interesting new approaches, I think.
Great. That's good to hear. That just dispels some of the notions of the conservative clinical world. And you've cracked that nut, which is fantastic. So we do have a couple of questions in the chat. So one is from Andrea Clermont, who says, one question to Marina: how do you validate the method of late slope activations in Brugada patients? Comes back to exactly this point of, revising your estimates based on clinical sensibility or intuition, whatever. So how do you actually validate, which of the negative slopes to pick?
Yeah, so if you're looking at activation, the Medtronic software, at least in the data sets we got, gives you an activation wave. So if you assume that to be right, then you have at least a ground truth to deal with. But then that will also have the same issue that it might pick just the wrong one. So there is no actual validation for that. And I think that is the main problem with ECGI, that, it depends so much on your filtering, on your signal quality, that is difficult to validate, and repolarization is worse because Medtronic don't do repolarization maps at all. Mm-hmm. And so when I computed it, it's like, is this what you expect? Is this not what you expect? And that is why it's important to have many beats. I normally deal with 3 to 5 beats per pacing modality or whatever the clinicians are looking at, and more patients, as many patients as possible, because that way you're kind of sure that, or not sure, but confident that the differences you're seeing are not specific to that one patient, or they're not caused by the data analysis you're doing. So for instance, for the Brugada syndrome patients, we're looking at repolarization as well, and we see pretty much the same distribution of repolarization times in all patients at baseline and follow-up. 3-6 beats, they're all the same distribution. So there are differences between beats, there are differences between patients, but the overall behavior is similar. So it's like, okay, then I'm confident that what we are seeing is actually the truth. For pacing, actually, it was easier, because in pacing, the activation waves and the repolarization waves seem to follow the same distribution. So that was easier for the clinicians to tell me, okay, I think this is right. Mm-hmm. While for the Brugada syndrome patients, we didn't have that luxury, because it's an abnormal disease, both in activation and repolarization, so they behave differently. So that was a difficult thing to deal with, and that is why we waited to have all the data from all patients and then do the analysis. While with the pacing data I got from St. Thomas's, I analyzed one patient at a time, because even within one patient we were able to tell if that analysis was reasonable or not. Mm-hmm. So that was Yeah,
that makes sense. I mean, we have conditions in which or cases or types of cases that, for which there's a certain intuition built up. So if we show that to the clinicians, they can quickly give us feedback. There are other times when we're showing them data they've never seen before, whether that's data from an AI algorithm or a repolarization map that they're not used to seeing. Then it's an education process on both sides. You really need a strong commitment, it sounds like, from the clinicians to also learn their way into it as you're learning your way into it.
Yeah, and also, the clinicians, at least in my experience, rarely cared about the maps. They care about the metrics to extract from the maps, because that's what they're going to publish. Mm-hmm. If the metrics are consistent between the beats, even though there are maybe some areas where the repolarization times, it doesn't look right, they're like, well, the metrics they're extracting looks right, is reasonable. We don't care that much about the map. So the metrics made more difference for me when dealing with the clinicians in general. Once we agreed on what they wanted to extract from the data and what they were interested in, then that was it made the collaboration much easier.
Yeah, yeah. Excellent. Thank you. So we have a question from Najiba, and I'm not quite sure. Najiba, you there? Do you want to unmute and pose it? I'm not sure I understand the question as you've typed it. So
I do.
sure.
I do actually.
Oh, you do?
I do
Good.
understand,
Okay, go
yes.
ahead. Go
yes,
ahead.
I understand. So, the protocol is the following. We pace from sequentially from over 40 different sites in the left and right atrium. So for each pacing site, it may or may not induce, we may or may not have a target. So once you go through all the pacing sites, majority of them will end up in rotors that are in the same location. So then we have a hierarchy of targets. The target that has the most pacing sites from which this rotor is induced is number one in the ablation targets. They go there first. Then we might have pacing sites that are the only pacing site that induce somewhere. So these are down in the list. So that's how we do it. So for each pacing site, we look at the target and then find what is the distribution of these targets and where they overlap. So that's the question.
Okay.
So I have here a question whether I see the need for personalized electrophysiology or whether anatomy would be enough. This is, of course, an excellent question. I don't want to do anything invasive. No. So I want to, whatever I can acquire, I would like to acquire it non-invasively. Here, ECGI might be of help to personalize. So we did actually a very extensive sensitivity analysis when you change action potential duration and conduction velocity to see how your targets change. We have a paper published on that. And I think in about 94 to 96% of the cases, the targets were the same, regardless of what was the action potential duration. But they were the same final target set. What is important is for one action potential duration, you might have a target in one place. And then when you repeat, because we do numerous repetitions, you ablate here, another target comes here. For different APD, this might be the first target. So the way they occur in the set is different depending on what is the action potential duration and what is the conduction velocity. But at the end, when you have a final target, so there is a relationship between electrophysiology and anatomy. But when you eliminate one of the vulnerable areas, like you don't generate there, they become more constrained, and in the end you catch everything, regardless of the action potential duration or conduction velocity. But that was in, as I said, I believe 94 to 96%. So we have about 3-4% in which there were new targets that we didn't capture. So that's the reality of it so far. What we are doing is our action potentials are now being created with genetic algorithms from patients with persistent, paroxysmal, or they are from biopsies. So we want to update that and see whether we almost finished with that paper to see whether there is a difference. So I am going for what I called a cohort specific rather than patient specific. So persistent, we've done that for ischemia in our VT studies. We did different patient diseases and different diseases, and you measure that, and then you have like a restitution, conduction velocity, action potential duration, activation recovery intervals. We create that, you do a genetic algorithm, and that's what we did. So we are going to repeat that for the atrial stuff. It's almost done.
Great. You have another question there. What do you think this is from Hannah Smith. Yep. What do you think the upper limit is for prediction accuracy of sudden cardiac death on your data set with current inputs? Could this be increased by increasing inputs? And if so, what data would you want to add?
Well, first of all, I would like more data. The problem with LG-MRI is it's not widely available. And so for us, it took us 6 months just to locate and figure out how we get the data. So I would like to have more data sets, of course, and so bigger cohorts. Now, I think that there are other things we might be predictive. Of course, ECG is a cheap tool that could be used in these things. Many people have done AI work on ECG. For me, I see the role of ECG in a very different way. I think ECG can be used to predict which of the patients have scar, and only then they do an MRI, because MRI is expensive. So if you can do so we just wrote a grant on that got funded. It's like you do AI on the MRIs to tell you whether the patient has scar, because you don't know whether the patient has scar. If they have scar, then you incorporate it in the, so that's our new way we are going with that.
That makes sense, because I wondered, listening to you, sort of how similar is your approach to these physically informed neural networks that we hear about a lot in the world. This
so
AI
I
world.
hired one of the best guys in those pins. He works with he was developed around
with
with
Carniadakis. Yeah.
So he is George Carniadakis' most successful PhD student. He's coming in August. I have a lot of projects for him, so we will see how that works.
Yeah. And I guess there's a follow-up question, which I think you're sort of addressing in bits and pieces, but how broadly do you see this whole merger going forward? How do you see the trend of using AI technology in precision cardiology? So where do you see that all leading?
I mean, it's got to be there. I think it's not going to be, doctors are saying sometimes, oh, we will be left behind, there will be no decision that, we are not going to be part of the decision. No, doctors who do not do AI will be left behind. Doctors who do AI will not be left behind. So to me, it's a process. Everybody is going to learn. Everybody is going to come on board. This will become a reality.
Yeah. I mean, we work with radiologists here, and they're very excited about AI, even if it seems like a risk to their profession, because they say some large percentage of cases don't really need my expertise that I've built up over years.
Well, good. Then you specialize in more complex cases and help more patients.
Exactly. That's exactly the perspective. Is if we could pre-screen with an AI algorithm a lot more of these straightforward cases, then I can really have the time I need to focus on the complex cases. And I think, electrocardiologists see it the same way when they're thinking about ECG analysis. They've used computer-based analysis for many years. So that they're kind of used to that paradigm of getting a first read from the computer and then glancing at it to make sure it makes sense with their intuition. And now what we're seeing is a replacement of that rule-based approach with a machine learning-based approach that hopefully will improve those early predictions, but not leave them any more vulnerable or less busy than they are now. All right, great. Well, I think we've taken enough of everybody's time. Thanks. This was really well attended. I'm so happy so many people showed up. We had well over 80 people watching. We'll put together the videos of this and make sure our speakers are comfortable with putting those on the website. But thank you all very much. The one technical meeting I can sometimes get Natalia to attend is computing cardiology. So let's hope we see a lot of you in Tampere this fall, and maybe we'll convince her to come. And I'm sure we'll have lots of material for some really exciting discussions, related to this topic and many more. So thank you all for attending.
Thank you all very much.
Have a great rest
Thank
of your day.
Thank you for having me.
All right.
Thank
Bye-bye,
you.
everybody.
Bye.